Hello, and welcome back to Mechanochemistry Discussions. This seminar series is hosted by NSF-funded Center for Mechanochemical Controlled Chemistry, or CMCC. I'm Mateusz Mariański, one of the center's investigators. As usual, the goal of this seminar series is to bring our community together. The seminar streams live on the third Thursday of every month at 11 a.m. as Eastern. They're also available to watch anytime on the YouTube channel. Also on YouTube, you will find all of our previous speakers as a part of the series, and I encourage you to check them out. We also have some excellent speakers planned for the upcoming months, and we hope that you will join us for all of them. Before we get started, a few quick thank yous. First, Dr. James Batias, the Center's Director, Jennifer Belcik, the Center's Administrative Coordinator, and two CMCC students without whom the seminar would not happen, Katie Floyd and Ryan Kwok. Please do continue to follow us on YouTube channel at CMCC Mechanochemistry Discussions. Before we start, a few quick guidelines. The seminar is being recorded. If you have any questions during the seminar, you can either email them to us at cmccdiscussions@gmail.com, or you can post them directly into the YouTube channel. Either way, they will be propagated to the speaker at the end of the seminar. Without further ado, it is my pleasure to welcome today's speaker, Professor Hao Yan. Hao Yan is an assistant professor at University of North Texas. He obtained his PhD from Harvard in 2010. Before joining UNT, he was a postdoctoral fellow and research associate with Stanford University and SLAC National Lab. His current research interests include high-pressure chemistry, material science, and biophysics. It is my pleasure to welcome Professor Yan to Mechanochemistry Discussions.
All right, everyone. Let me just say that it's a great pleasure to be invited to this seminar where I can tell you a little bit about our work on mechanochemistry in the compressive regime on molecular compounds. Let me just say that I've viewed some of the previous presentations, and I saw a lot of familiar names, prestigious names. Wendy Mao was here. She ushered me into the high pressure field. Steve Craig, that I've met, although just a few times, taught me so much about mechanochemistry. So I'm very much honored to be part of this series. All right, so let me begin by stating the obvious. We have all sorts of interactions between matter and pressure. This goes from a gentle touch of the fingertip all the way to center of planets, right? For me as a chemist, I'm mostly interested in the pressure range of megapascal to gigapascal. And this is because if you calculate the PV work associated with these pressures, we're talking about kilojoule per mole kind of energy. So this corresponds to the energy needed to change conformations of molecules or form or break chemical bonds, i.e. the realm of molecular chemistry. So what can we do with pressure? Well, you can look at it from different perspectives. If you think about it as a phase transition problem, of course, right? There's this vast range of exotic materials that you can explore when you raise the pressure, like one of them being diamond. Actually, the synthesis of diamond is one of the driving forces that initiated whole high pressure research. But more recently, people have found that you can make say insulating sodium, metallic hydrogen, close to room temperature superconductors, all these wonderful and exotic materials. Now, let me show you one example that I studied when I was a postdoc. We're looking at the high pressure behavior of this molecular compound. This is a tetrameric copper thiolate compound. On the right here, I'm showing you the pressure dependent X-ray diffraction pattern. And you see that it actually is pretty boring, right? You see, you have all these peaks that are shifting to the right, which means that the unit cell is shrinking, and up to a certain pressure, the lattice collapses and everything turns into a flat line. What's actually interesting about this system actually goes into the molecule. In this particular one, we're actually seeing that the bond angle around this otherwise linear copper actually starts to be bent. under pressure, and this drives a charge transfer that eventually leads to a reduction of copper 1 to copper 0, solely driven by mechanical force. Right? So this, I think, is a wonderful example that shows that when we consider the high pressure chemistry of molecular compounds, the details really lies not in the long range order or the lattice, but rather in the local structures within the molecules. All right. So why high pressure chemistry? Why do we want to use pressure instead of heat or electricity or light as our driving force? Right. I want to take a step back. Mechano or no mechano. Let's think about just chemical reactions, and I want to chart them on this map where I have the entropy of the reaction on the x-axis and the energy level on the y-axis. Right. So you would have something that's extremely chaotic, low selectivity, and low energy, such as the carbocation rearrangement. Right? You can have explosions and combustions on the upper right corner. These are very high energy reactions, but also very chaotic. What I would argue is that the really interesting chemistry and the challenging ones lies in the upper left corner, where you want to drive extremely energetic reactions. For example, the cleavage of nitrogen double bond, but with very low entropy. Right? Low entropy in this sense means high selectivity, high specificity, and high efficiency. Right? Apparently, nitrogenase and other enzymes learns how to do this long time ago, and there's a lot for human beings actually to catch up in terms of technology. And this is where really a lot of the chemical challenges that are associated with society, energy, environment are lighting, CH activation, CO2 abatement, so on and so forth, PFAS reduction. These all lies in the corner where you need to drive the reaction with very high energy, but with relatively low entropy or disorder. Okay, so now let's think about what pressure and temperature does. This is, I promise, the only equation I'll show in this talk. Temperature, of course, increases the energy of the system. It also increases the entropy of the system. Right? This is the third law of thermodynamics. Pressure, if you think about it, in general, if you especially if you're dealing with a solid, compressing it increases the total energy of the system, but it also, for normal materials, decreases the entropy of the system. Turns out the entropy slope versus pressure is the opposite of the thermal expansion coefficient, which in most materials is a negative number. So this, to me, suggests that exploring the high pressure regime of reactions really allows us opportunity to go to this higher top left corner, the high energy, low entropy regime, again, translating to high specificity, high efficiency reactions. All right, microscopic picture of a pressure-driven reaction. This is kind of similar to all the other genres of mechanochemistry, right? We're talking about adding a F delta X or P delta V term to your energy landscape. This results in a tilting or modification of the PES, which then favors new ground states and the new reaction pathways. As a chemist, I like to think of it as having a force or pressure pushing my molecule up the slope of my energy barrier, like this teeny tiny stick figure Sisyphus pushing a rock. And in our cases, the Sisyphus would not have done work in vain. The molecule can be pushed over the barrier and resulting in a new reaction. So then what's the difference then between, say, a tensile stress-driven mechanochemistry that you can do by just putting on the polymer or sonication, or in other forms, mechanical grinding that gives you a friction and a shear-driven reaction. In these cases, the stress, which I represent here by a stress tensor, is directional, right? And the directional stress usually leads to an anisotropic strain. This could mean stretching of a chemical bond or opening up of a dihedral angle, which eventually leads to reaction. In our case, we're using hydrostatic pressure, which is another way of saying the pressure is static and is uniform in all directions. Experimentally, this is generated in a diamond level cell, which I'm sure a lot of you have heard about from previous talks in this series. So the stress is isotropic, okay? But we have to remember that most molecules that we deal with are intrinsically anisotropic. in their compressibility. So we're really relying on the intrinsic anisotropy of the molecule to determine which direction does this isotropic stress induce a strain. And in cases where the strain aligns with a particular reaction coordinate of the molecule, there we go. We have chemical reactions. And as I said, experimentally, this is done with a diamond novel cell. For those of you that have not heard about this device, this is two pieces of cone-shaped diamond with a broad base and a sharp tip, sandwiching some materials in between. And pressure equals to force divided by area, right? So a very small area translates to a very large pressure. So just by tightening some screws, we can generate gigapascal level of pressure. And because diamond is transparent, we can shine all kinds of magnetic waves into our sample and do in situ spectroscopy. So this is a great tool for mechanistic studies. All right, so I think I've thrown enough of these lofty ideas. Let me show you a few concrete examples. Okay, the first one I'll show you is a topochemical polymerization of conjugated diacetylene. This is a reaction not entirely new. People know that if you put conjugated diacetylenes together in a solid crystal, you can drive the reaction by heating, by light, or by pressure. And the guiding principle here is the so-called topochemical selectivity. It means that the active sites that get spatially close to one another tends to react together and form new chemical bonds. In this particular case, we're looking at a so-called 1,2 addition, which means that the carbon 1 in the first diacetylene attacks the carbon 2 in the adjacent diacetylene, forming a new bond and eventually this polymerizes into a polyacetylene kind of structure with a triple bond as the side group. Now, one of the twists that we did here that's different from the literature is that we're doing this reaction in a two-dimensional confinement. We have collaborators that synthesize a two-dimensional perovskite with the conjugated diacetylene as the organic spacers in between the lead bromide layers. So this is what we expect, and this is what we see in experiments. We can put a single crystal of this 2D perovskite in the diamond level cell, compress it. We can see the color change, but also in situ Raman spectroscopy, we can see the rise of this new band at 1600 that corresponds to the stretching of the CC double bond. Now, once this polymer is formed, it is metastable. We can release the pressure, recover the material, and you can actually see that the content of sp2 carbon actually rises by this pressure. Now, there are two things that's particularly interesting to us about this reaction. The first one is how does pressure drive the reaction? Right? As I said before, the principle here is that whatever strain you're inducing the molecule, it should push the structure closer to the transition state. Right? This is known as the Hammond postulate or the Bell theory. But how does it happen in this case? Well, we modeled the transition state for the initial dimerization of the two adjacent diacetylenes. And what we found is that it takes two components, right? There's the spatial adjacency or vicinity of the C1 and C2 of these two adjacent diacetylene molecules that's needed to form the chemical bond. But also you have to realize that in forming this bond, the hybridization of the carbons goes from sp to sp2. In other words, the bond angle goes from 180 to 120. So there's also an element of the bending of the bond angle here. And that's where the two-dimensionality of this particular system comes in, right? Looking at the structure, this is apparently a very mechanically anisotropic system. The in-plane compressibility governed by the covalent and the ionic lead bromide bonds extremely large, extremely small, right? So this is a very stiff in plane, but out of plane, there's only van der Waals interaction. So the whole crystal is much more compressible in the z direction, so to speak, the out of plane direction, even if you're applying a isotropic pressure to it. Okay. Now, what does that mean? It means that the molecules, these otherwise linear diacetylene molecules, actually got bent under our hydrostatic pressure. And that bending, again, drives the ground state structure of this molecule towards the transition state, which requires somewhere around a 140-degree carbon-carbon bond angle. And that, we believe, is what lowers the activation energy and allows this reaction to happen at room temperature. That's item number one. Number two, now once these polyacetylene molecules form, or polymers form, we still have half of the triple bonds that are not reacted, and in principle, they can react, and they can go undergo another step of polymerization, forming a polyacene or a single file graphene nanoribbon, if you will. Right? What we see is that this reaction does not happen under high pressure. You can see that very clearly from the color change, right? If I press it to 13 gigapascal at room temperature, I got my polyacetylene, and keeping that pressure and heating it up, nothing changed. If I release the pressure, heating it, and all of a sudden the polymer becomes black. You can also see this from the relevant content of the sp2 carbon going from compressed and heated at high temperature, at high pressure, not much change. But if you release the pressure and heat it, then all of a sudden the sp2 carbon amount increases by a lot. Okay? So what we think happens here is this. Now, in order for this reaction, the second polymerization to happen, these two triple bonds would have to rotate into the same plane in order to form this polyacene structure. And that rotation, we believe, increases the thickness of the organic layer, which goes against the pressure, right? You have to pay a very large energy penalty for these two triple bonds to rotate into the same plane. In other words, pressure in this case is limiting the accessible conformational space of our intermediate product, and therefore preventing the reaction of the second step. Now, also interesting here is that if we just take the pristine material and heat it directly at room temperature, at room ambient pressure, we see that the product is likely a mixture of the polyacetylene and polyase. In other words, by using pressure, we're encoding a unique selectivity in this reaction. So tying into what I was talking about in terms of driving high energy and low entropy reactions. I think this is a wonderful demonstration of that. Now, what is good of this material? Well, it definitely shows some interesting shift in its PL spectrum. We're also exploring the possibility of using this compound as a high efficiency photovoltaic material. Okay, let me tell you another story. This one goes to a metalloorganic or a coordination compound. Here, what we're interested in is to try to capture some of the transient species in these catalytic cycles of copper proteins. Okay, shown here is a pyrosinase, but this also happens for PMMO, catechol oxidase, so on and so forth. What we came to notice in a lot of these works is that although the resting structure of these compounds usually involves 2 copper and a bridging oxygen species sitting in a symmetric structure, the active site or the reactive intermediate or the transition state usually features a highly anisotropic structure. Looking at this one, right? This copper has coordination number 4, this other copper coordination number 5. So this seems to imply that such breaking of symmetry induces certain kind of enhanced reactivity. Okay, this has been seen actually not only in enzymes but also in artificial catalysts such as the copper activated zeolites for methane oxidation. The challenge here is that capturing these transient structures is remarkably difficult, and trying to synthetically access these kind of asymmetric structures by conventional means is also quite challenging, right? You're talking about installing 2 sets of dissymmetric ligands on two otherwise equivalent or chemically equivalent copper centers. That is a rather challenging task. So comes in high pressure. This is my the work by my postdoc Pete Manata, who's on the job market right now. He looked at the high pressure behavior of a dicopper mu hydroxyl compound. So we have 2 coppers here bridged by a hydroxyl group and supported by TMPA ligands. The starting material is all symmetric. Okay? We put it under a pressure of about 8 to 9 gigapascal. What we have seen is that there's a color change going from blue to greenish. We can trace the structural changes by Raman spectroscopy. For example, you see here that the stretching mode of the OH bond remains stationary against a otherwise universal blue shift of other vibrations due to mechanical stiffening of the chemical bonds. And at the same pressure, when the color change happens, this vibrational band disappears. We can also see that the breathing mode of the pyridyl rings changes profile and going towards a pyridinium or a pyridyl plus a proton. Right? So combining these evidences, we think that what happens is that the pressure drives a intramolecular single proton transfer. So the hydrogen on the hydroxyl hops off and goes to one of the pyridyl arms of the TMPA. That arm then dissociates from the copper and generating a compound like this. We can actually model these structures and obtain a stable structure using DFT. Now, the interesting thing is that this compound, once generated, is actually metastable. We can take it out of the diamond cell, keep it in the glove box, it stays there for a couple of days. So this really allows us to do a lot more characterizations. Let's talk about the mechanism here first. What we think happens, and this is supported by DFT calculation, is that the uniform isotropic pressure generated in the diamond cell, again, induced some highly anisotropic deformation in these molecules. In this case, it's bending the copper-oxygen-copper bond angle, and that bending weakens or decreases the covalency of the OH bond while bringing a pyridyl arm closer to the hydrogen, and this leads to a proton transfer. We can analyze this by looking at atomic distances and charge distributions, as well as spin states, which I would not go into all the details. But as we have found, probably the most striking thing about this is that this proton transfer is not electronically innocent, let's say that way. We started off with a compound with two copper-2 centers, and after this transfer, you see that the two coppers now sit in different ligand environments. In other words, we break the symmetry of the molecule, and that broken symmetry leads to a mixed valency. We start to see that we have a mixed copper-1 and copper-2 species with a ratio of 1:1. So what we think, and this is again backed up by CAS-SEF calculation, is that the copper that sits in the 6-coordinated field is still a copper-2. The 5-coordinated field is still a copper-2, whereas the other copper that now sits in a 4-coordinated system is a copper-1. And that implies that the bridging oxygen now should carry some radical characteristics. So it's probably a mixture between oxo and oxyl. And being that oxyl nature dictates that this bridging oxygen is just hungry for hydrogens in terms of hydrogen atom abstraction reaction. We can demonstrate this by the hydroxylation of a sec butanol that's co-crystallized with our molecule inside the crystal. And we see that the oxidation of the sec butanol generates ketones and diols with pretty high yield. Probably this is really a tour de force experiment performed by Pete. He built a device that allowed him to load methane into the diamond cell, compressed the crystal plus methane system up to about 15 gigapascal, and he recovered a liquid after the compression and release of the pressure. Now, analysis of this liquid showed that it actually is methanol. So in other words, once we break the symmetry of this dicopper center, the bridging oxyl is so strong a oxidant that it can oxidize the CH bond in the methanol with a BDE of up to 105 kcal per mole. Are we saying that every flaring ox field or oil field in Texas should have a high pressure device on top of it? Probably not, right? But this is really to us informative in how we can design catalysts and other chemical structures that use asymmetry as a new tool or a new way of thought in terms of activating these otherwise really difficult to activate CH bonds. In fact, this is not the only example that we're interested in the copper oxo sort of regime, right? There are all kinds of interesting reactions. For example, we have been looking at the cis-peroxo to trans-peroxo transition in dicopper dioxo species. This represents some of the early stage binding events in, say, tyrosinase and hemocyanins. But I will not go into all these details. Instead, I want to show you a case where pressure doesn't carry the day, right? In other words, you really need pressure and heat to drive the reaction. In this case, our model system is a isomerization in a transition metal dinitrogen compound, which again is tied to the nitrogen activation, one of the arguably most challenging reactions that has significant impacts on human society, energy, environment, so on and so forth. Right? The model compound that we identified by some computational screening was reported by Paul Chiric back in 2014, and it features a dizirconium dinitrogen structure. The zirconium is ligated with CPs and alkyl ligands, and the nitrogen binds in a so-called end-on conformation. So one nitrogen binds to one zirconium, the other nitrogen binds to the other. Our DFT calculation suggests that at high pressure, the dinitrogen can turn 90 degrees and it forms a so-called side-on conformation. So each zirconium now binds to two nitrogens. Right? This switching can be seen in our analysis of the atomic structures. Computationally, we also predict that the nitrogen-nitrogen stretching mode would show about 200 wave number redshift when this switching happens. Okay? So the DFT calculation only tells us that there is this possible new ground state at high pressure lying out there. It doesn't really tell us how to get there, right? So my graduate student Cindy went on to do the experiment. She synthesized this crystal, loaded it in a diamond enamel cell, put it under pressure, and then measured the Raman spectrum. This is a busy slide, so let me walk you through that. Starting from the lowest pressure here, 0.1 gigapascal, we're increasing the pressure, and you're seeing initially this blue shift of the nitrogen-nitrogen stretching mode. This corresponds to the mechanical stiffening of the bond. And up to about 60 gigapascal, you see a new band rising. This is about 50 wave number redshifted towards relative to the initial peak. We don't exactly know what this species correspond to. It likely is a new conformation stabilized by pressure, but we're pretty sure that this is not the side-on compound that our DFT predicted. Right? As you increase the pressure, you see that this new compound peak becomes stronger and stronger. But in the region where we expect to see the vibrational mode corresponding to the side-on dizirconium dinitrogen, there's no change in the spectrum. It's all just a flat line. Okay, and when we release the pressure, the molecule just returns to its initial state. So whatever it is transitioning and forming under pressure, it doesn't really give us the side-on structure as the DFT predicted. We can see the same thing using N15 instead of N14 for the experiment. All right, so we've been doing this for about a couple of years and got really frustrated in the puzzle as for why we're not getting there, even if we're putting a ton of energy by compression into this molecule, until one day Cindy said that, well, let's just try some heating. Right? So she compressed the molecule or the crystal in a diamond enamel cell to about 10 gigapascal, put it on a hot plate, heat it to a few tens of degrees, and then measure the Raman again. And all of a sudden, the magic happens, right? We're seeing this peak corresponding again to the side-on zirconium dinitrogen arising as a function of temperature. This can be seen somewhat in the N14, but much more clearly in the N15. You can see the rise of this band around the 1450 wave number, which corresponds again to the side-on compound, right? So we're trying to understand what happens here. Apparently, the pressure doesn't really push the reaction all the way to the endpoint where we expect things. So we started to do some DFT modeling. The first thing we calculated is the reaction coordinate. We used the nudge elastic band or NAB method. And what we found is that this transition actually has two settle points, starting from the side-on structure undergoes the first transition going to what we call a triangular structure, right? So this this zirconium binds to one nitrogen, the other zirconium binds to two nitrogens. And from here, there's really a shallow barrier that transits to the side-on structure. Okay? Now, if you dissect the reaction coordinate here, what we realize is that it has actually two components. There is the shrinking of the zirconium distance, right? But there's also the rotation of the nitrogen species relative to the zirconium, which we capture here by the zirconium nitrogen vectoral angles. So it goes from almost 0 degrees at the beginning to close to 100 degrees at the end. Okay? So you need 2 motions in the molecule for this reaction to happen. We put this 2 components of the reaction coordinate on the x and the y-axis and the energy on the z-axis. So what we're looking at here, the red line here is the energy profile along the reaction coordinate. If we think about the effect of pressure, it can be assimilated or approximated as just the shrinking zirconium distance. So we're also taking a cut of the PES along the straight axis, the shrinking zirconium. So you see that without turning the nitrogen relative to the dizirconium axis, this slope becomes extremely deep and the barrier is extremely high until when it reaches some absurdly high energy, then it falls back to the intermediate triangle state, which then proceeds to the Sidon. Right? Here I'm showing you the change of the structure along the strain axis by pressure versus the reaction coordinate that's actually needed to drive the reaction. So what we're seeing here is that pressure only pushes the zirconium distance closer together, but it does nothing to help with that rotation. In other words, the strain axis driven by the pressure in this case is misaligned with the reaction coordinate. And in this case, the pressure doesn't drive the reaction all the way to the product. You also need a thermal activation that assists the rotation of the dinitrogen in order for this reaction to happen. All right, as a supporting evidence for this mechanism, we also see some interesting inverse kinetic isotope effect. Again, the reason being is governed by the Hammond's postulate, right? Your transition state structure is more similar to the product with a higher energy. In this case, the product here is the triangular structure with 3 zirconium nitrogen bonds, whereas my starting structure, the end-down structure, has 2 zirconium nitrogen bonds, right? So replacing N14 with N15 would decrease the 0-point energy of the transition state more does it to the starting structure, the end-down, and therefore we expect the N15 structure to have a lower barrier than the N14. And indeed, we see that the N15 compound transits to the side-down structure much faster than the N14. All right, so let me try to wrap up here and give you some prospect of what we think can be done with this molecular mechanochemistry in the compressive regime. And one thing that I'm really interested in is to use this as a fundamental tool for mechanistic studies. Okay, going back to the very nature of chemical reactions, right? Chemical reactions can be appear to be very fast and violent or very slow, but on the molecular or microscopic scale, elementary reactions are always fast, right? You have your molecule vibrating at its reactant ground state, and once it gains enough energy, it hops over this barrier, hop to the adjacent ground state, and that process is really on the time scale of femto to picosecond. Now, the standard and canonical go-to method to study this is by optical pump probe, right? The caveat here is that for optical pump probe, you're really just kicking the molecule up to its electronic excited state, which then finds its way to relax back to the electronic ground state and arriving at the product structure. So one can argue that the reaction path taken by this pump probe process is fundamentally different from what a thermally activated reactions would look like, right? And there are reactions that doesn't need or doesn't take any optical excitation. For example, a lot of the reactions going on in our biological system, enzymes, right? A lot of these reactions doesn't have anything to do with optical excitation. So what we think might be possible is that, again, going back to this modulation of the potential energy surface using stress or using hydrostatic pressure in this case, we can push the ground state molecular structure along the reaction coordinate and allowing it to climb the energy barrier, just like shown here. Now, if we do this with high pressure or variable pressure and cryogenic temperature such that we freeze the vibrational the molecule to their vibrational ground state, then we should be able to quasi-statically map out the structure and possibly energy of the reaction path. Instead of having the molecule just dashing over the barrier, we can take it for a casual, quasi-static walk or a traversal along that path. And in doing so, we can use steady state spectroscopy tools to really understand the structure. Now, this hopefully will translate to a fundamentally new way to study reaction mechanisms and renew our understanding of the nature of chemical reactions. Gonna just show one example here. The system that we've been looking at, this is the so-called side-on peroxide to bismuth oxide isomerization, again in the dicopper center. This happens as a key transition in the catalytic cycle of tyrosinase, catechol oxidase, and all other sorts of binuclear copper enzymes. People know that this exists. We know that this has to do with their catalytic activity, for example, transforming phenol to catechol and to quinol. We don't really understand how this process happens, especially we don't understand how the nominal oxidation state of the copper changes from copper 2 in the starting structure to copper 3 in the end structure. And this is something that's, as far as I know, has not been studied by optical pump probe, primarily because there's really no optical excitation that's associated with this process. But this is something we can possibly study by taking a molecular analog of the enzyme, using pressure to push the copper centers closer together and observe the structural change along the pathway. All right, so with that, let me just stop here. I want to thank the organizer and CMCC again for inviting me. The people that really made this work possible, my graduate student Cindy, postdoc Pete, undergraduate David, and my former master student Lama, they really contributed the majority of the work that I'm showing you here. I thank the funding agencies and the facilities that make the work possible. And I'll pass the floor back to you for questions and comments. Thank you.
Thank you very much, Paul, for the very interesting presentation. I think we have several questions from the audience. So the first question is going to be that a lot of the studies that you presented appear to require a very carefully designed force acting along specific coordinates, and this required this anvil cell setup. Can you imagine that similar forces can be active or can be activating this catalyst in the ball mill reactors or something that can be acting on the larger scales?
I would believe so. Now, I would dissect this problem into these questions into two parts, right? One is a different type of force, right? The essential thing that the diamond cell provides is a perfectly hydrostatic environment, right? Your pressure is guaranteed to be isotropic from all directions. Now, if you switch that to, say, a grinding force or sonication, milling, it adds interesting things, right? Your stress tensor, as I show at the beginning of the talk, is definitely no longer diagonal. Now, what does that do to the molecule? I think you have to analyze that in a case-by-case manner. There are definitely cases where we see adding a anisotropic element to the stress actually assists with the reaction. On the other side, the other part of the question is scalability, right? And yes, diamond cell is not your go-to for any synthetic chemistry. There are devices like the large volume press that are designed for, say, the gemstone industry, and they can produce grams scale of samples with a similar kind of pressure that we're using. So to some extent, the scalability issue is not quite solved, but not as bad as some of you would think, depending on where you look and what kind of apparatus you choose to use for these problems. I hope that answers the question.
I hope so too. So the next question is about the computational techniques that you use to study these problems. And I must say that they were pretty involved. Could you comment a little bit more about the details of your of the functionals that you use and for studying and basis sets for studying these radical states of the metals after all?
Sure. Again, depends. We typically use two types of tools, right? For any modeling of crystal structures, we go to quantum espresso. So this is a plane wave based basis set, right? We're pretty much practically limited to GGA level of theory, PBEs. For say correlation effects, we just use the Hubbard potential to account for that, and that seems to work pretty well. Now, for molecular studies, for example, for this one, I believe we used a BP86 basis set, and I want to say a TCVP or DEF2 TCVP. Sorry, the BP86 functional and DEF2 TCVP basis set for these atoms. It really is a trial and error case. I would say our standard is to register the calculation results or gauge the calculation results against our experimental findings. For this particular system, for example, the zirconium system, the metric of importance is the vibrational frequency of the dinitrogen stretching, and we choose the functional that best matches our experimental data. For the previous dioxo system, important thing involves, say, the correct prediction of the copper oxo stretching mode, and those are the things that we use to gauge our calculations.
Okay, here's the next question. So in the introduction, you discussed entropy a lot. Have you ever measured changes in the entropy in your process?
That would be ideal, but we have not. I would have to say we have not. There are ways to do that. Thermometry or calorimetry is to me the go-to method for actually measuring the entropy. Most of the devices so far are designed for close to ambient conditions, but I know that there are developments in the high pressure community towards doing thermometric experiments in the diamond level cell, and that will be tremendously useful for understanding the mechanisms. So yeah, possible, but we haven't gone there yet.
Mm-hmm. And the last question that we have is, does your data show that pressure always increases energy of starting material, but does not always lead to a faster reaction?
I would say so, yes. For example, in this very early work that we did, you can actually just take the diffraction patterns, do some fitting, find out the volume of the unit cell, and then fit that to, say, a third order Birch-Morgan equation of state, right? Then you can, from there, calculate the energy change as a function of shrinking volume, cell volume, or increasing pressure. Yeah, pretty much in all the cases, we find that the energy goes up. So here's the key point, right? We're not just isotropically dumping the energy to every degree of freedom of our molecule, unlike, in the thermally driven reaction, right? So there you're governed by the equipartition theorem, right? Here, our energy, for lack of a better word, goes in certain directions. We know that energy is scalar. It doesn't have a direction, but it strings certain internal coordinates or degree of freedom in the molecule. And the key, as I shown in that dizirconium dinitrogen case, is that you have to align this string axis with the reaction coordinate. Right? Otherwise, you're pushing the molecule up a very steep slope that really doesn't generate any new Brown states or doesn't allow you to reach a new Brown state.
Mm-hmm. Okay, so these are all questions that we had. So once again, Hao, thank you very much for your seminar. And thank you everyone for attending the seminar online. Thank you.
Thank you for having me.
Thank you, Professor Yan, for outstanding presentation, and thank you so much for joining us. Just to remind you that all of our previous seminar speakers are available to watch anytime on our YouTube channel, and I encourage you to check them out. We also have some great seminar speakers planned in the upcoming months, and we hope that you will join us for all of them. Thank you again.